Hello, my name is Qinjiang Zhu. It's my great honor to join the first International Electronic Conference on Medicinal Chemistry and to introduce our research about a method for testing drug-resistant HIV mutants. Here is the scheme for the method we propose. Briefly, the HIV protease gene from the patient will be cloned and expressed in the bacteria, and then the bacterial cell lysate will be used directly to do the enzymatic reaction with three modified substrates in the presence of antiviral drug. The products from the enzymatic reaction will be subject to the fluorescence reaction with catechol to generate the corresponding fluorescence products, and this product will be detected by HPLC. And finally, the IC50 of the drug for the patient-derived HIV protease will be calculated and compared with that from the wild-type HIV protease to evaluate the drug resistance. And this is the abstract for this study. I will explain the detail in the following slides. When we're talking about HIV, everyone knows that HIV infection causes AIDS, one of the most serious infectious diseases in the world. So far, there's no vaccine and cure for HIV infection, but we have dozens of antiviral drugs to prevent or delay the onset of AIDS. Based on the targets, this drug can be classified into 5 types as shown here. And the HIV protease inhibitor is one of the most important antiviral drug for HIV infection. It blocks the function of HIV protease, so the progenitor virus could not become mature and efficient. Until now, as we know, about 10 HIV protease inhibitor has been developed and used in clinic. But the problem is the growing drug resistance now is impairing the efficacy of this drug. According to the latest report from World Health Organization, a moderate level, about 5 to 15% of drug resistance has been observed around the world. So besides the new treatments for HIV infection, we also need the routine drug resistance testing for the patient to help to use this drug reasonably and then to avoid failure in the antiretroviral therapy and slow down the development of drug resistance. Currently, there are 2 types of drug resistance testing in clinic. One is genotypic testing, another is phenotypic testing. Genotypic testing looks for gene mutations that associate with drug resistance. But for the newly emerging mutation and the complicated mutation combinations, genotypic testing cannot predict the drug resistance accurately. Phenotypic testing measures the drug resistance directly. The clinically used phenotypic assay is also called the recombinant virus assay, and this figure shows the phenotyping process of one commercial available kit used in clinic. We can see that the virus gene was first amplified from the patient's blood sample and then cloned into the recombinant virus vector, and this vector was transferred into the host cell to produce the recombinant virus. After titration, the recombinant virus were used to infect the T cell again in the presence of antiviral drugs, and then the replication of virus was measured, and finally the drug resistance was determined based on the front change of IC50 when compared to wild-type virus. Obviously, this is a time-consuming process. Usually, it needs 3 to 4 weeks, and it also is an quite expensive testing because allow $800 need for one sample. So previously, our laboratory has reported that peptide with free N-terminals can react with catechol under a specific condition to form the fluorescence products, and this reaction has been proved to be a simple and cheap way to detect the peptide. So in this study, we set up an assay for drug resistance testing based on this peptide catechol reaction, and this figure shows the process of our proposed assay. As I mentioned at the beginning, HIV protease gene is first amplified from the patient's blood sample and colored into the expression vector, and then the vector is transformed into the E. coli cell after inducing expression. The cell lysate contain HIV protease was used directly to the enzyme reaction in the presence of HIV protease inhibitor. And in the reaction, 3 report HIV protease substrate peptide were used, but in the N-terminus of this substrate, ST glue was added to them and to stop the direct reaction between the substrate and the catechol. After the enzyme reaction at 37 degrees for 4 hours, this substrate will be cleaved by the HIV protease into 2 parts, and only the product with free N-terminal will react with catechol to form the fluorescence product peptide. And this fluorescence product was separated and detected and quantified by HPLC, and finally the drug resistance was detected based on the IC50 comparison between the mutant and the wild-type HIV protease. And this assay could be finished within 1 to 2 weeks, and it should be cheaper than the recombinant virus assay. So to prove this assay is workable, and the calibration curve for these 3 product peptides was first made, and here the synthesized product peptide were react with the catechol under the condition, and then the fluorescence product were analyzed by reverse phase HPLC. Figure A is the chromatogram showing the peaks of these 3 product peptides, and figure B is the calibration curve. Y-axis is the peak area, and the x-axis is the peptide concentration. We can see there's a good linear relationships between the peptide concentration and the fluorescence signal. And the detection limit for each peptide can be far as 5 picomole. This research suggests that the conditions for the catechol reaction and the HPLC analysis are suitable for the detection of these 3 peptides. This slide shows the preparation of wild-type HIV protease and its 2 mutants. So previously, the wild-type HIV protease gene has been coded into the expression vector in our laboratory, and in this study, 2 known amino acid mutations were introduced into HIV protease to generate mutant A and mutant B protease. And mutation G40A-V means the amino acid glycine at the position of 40A in the HIV protease has been mutated to the variant. And the V32I means the variant at the position 32 has been mutated to the isoleucine. The mutation G40AV has been reported to cause resistance to secondary, and one of the clinically used HIV protease inhibitors. And V32 has been reported to cause resistance to indinary and some other protease inhibitor, but not secondary. And the difference between the secondary is the reason why we choose this mutation for this study. And after the vector was made, they were transferred into the E. coli, and then expression, and then sonication, and then get the cell lysate. And the concentration of HIV protease in the lysate was roughly determined by Western blotting, and the standard is the commercial available HIV protease and wild-type and mutant A, mutant B lysate. So after quantification, wild-type HIV protease were first suggested to the activity experiment. The substrate mixture was first incubated with the lysate containing the wild-type HIV protease, followed by the catechol reaction and the HPLC analysis. In figure A, a different amount of wild-type HIV protease were incubated with the substrate mixture at 37 degrees for 4 hours, and in figure B, lysate contain 5 picomole of wild-type HIV protease were incubated with the substrate mixture at 47 degrees for 1 to 16 hours. We can see as the increasing of HIV protein amount or reaction time, the concentration of the product increase, suggesting that this assay could be used to detect the activity of wild-type HIV protease. To further test the assay, whether it can be used to measure the activity of HIV protein mutant, so we perform this experiment with if the lysate from the wild-type HIV protease and the mutant A, mutant B, and here the lysate contain 5 picomole of each HIV protease were incubated with substrate mixture at 37 degrees for 4 hours after chemical reaction and HPLC analysis. The result is shown in the figure A, and basing on this result, the ratio between the substrate cliff in each reaction was calculated and shown in the figure B. We can see this assay can detect the activity of HIV protein mutant, and also can show the different cleavage patterns of them on these 3 substrates. So the Km value measured by this assay show that the mutation lead to the different affinity of HIV protein to these 3 substrates. So obviously, this HPLC-based flow-metric assay can also detect and distinguish the activity of HIV protein mutants. And this slide shows the result of drug-resistant testing of mutant A and mutant B. Unlike the activity experiment shown in the last slide, the enzyme reaction here was performed in the presence of protease inhibitor. And figure ABC was the result from corresponding to secondary based on different substrates, substrate 1, substrate 2, substrate 3. And the figure D was the result corresponding to indinary based on the substrate 1. So the y-axis is the inhibition rate and the x-axis is the concentration of inhibitor. We can see when compared to the wild-type HIV protease, the bright line, the mutant A, the red line show a delay curve no matter which substrate was considered. So here you can see the delay line here. And when we look at the mutant B, the blue line, and we can find a delay curve only for the indinary but not for secondary. And no matter which substrate was considered, here we just show the result from the substrate 1. And according to this inhibition curve, the IC50 was calculated and shown in the upper table. So this IC50 is for the secondary, indinary, and from different HIV protease and basing on different substrate. And when compared to the IC50 for the wild-type HIV protease, we calculate the fold change in IC50 and so in this figure and this table. And here you can see, because we compare to wild-type, so HIV protease, so here is O is 1, and but for the mutant A and mutant B we can find and larger fold change in IC50 can be found for secondary from the mutant A but not for the indinary, and for the mutant B it only shows the larger fold change in IC50 to indinary but not for secondary. And actually so far we don't have enough sample to set up the cutoff value for this assay, but referring to the cutoff value set by the clinical use phenol-sense assay, we can make the conclusion that the mutant A is secondary resistant and mutant B is indinary resistant. This conclusion is consistent with the report phenotype, suggesting that this HPLC-based flow metric assay can evaluate the drug resistance of HIV protease. HIV protein mutants. And because to calculate IC50 need a series of dose response data, which means a relatively long time is needed. To shorten the detection time, a single inhibitor concentration experiment was tried. In this experiment, inhibitor was used only at a single concentration, and the drug resistance was determined based on the flow of resistance, which was calculated by these formulas. And here the A means the peak area of product peptide, and the I means the inhibitor treatment, and 0 means without inhibitor treatment, and WT is the wild type HIV protein, and the M is for the HIV protein mutants. And the single concentration here is the IC50 of the protein inhibitor for wild type HIV protein, and 4 clinically used HIV protein inhibitor was used to test this method. They are secondary, indinary, lopinary, and the litonary. And the result was shown in this figure. ABC represent the result from 3 substrates. We can see when compared to the wild type HIV protein, the mutant A, the middle one, show an increased flow of resistance to secondary, the black bar, no matter which substrate was used, and the mutant B show an increased flow of resistance to the indinary, the left bar, no matter which substrate was considered. So this result suggests the mutant A is resisting to secondary, and the mutant B is resisting to indinary. And both the mutant A and mutant B didn't show obviously resisting to another 2 inhibitor, lopinary and litonary. These results are consistent with the result from the IC50 comparison assay, and also consistent to the reported phenotype. It seems that this single inhibitor concentration method has potential to evaluate drug resistance of HIV protease more rapidly, but still need further experiment with more samples to confirm. In conclusion, in this study, a HPLC-based 3-substrate flow metric assay was set up for the activity and drug resistance detection of HIV protein. And this assay was tested with wild type HIV protein and its 2 known mutants under the treatment of 4 protein inhibitor, and it showed a consistent result with the reported phenotype. At the same time, a single inhibitor concentration assay was also tried for the rapid evaluation of drug resistance. Our study showed that this assay has potential to serve as a cheap, rapid, informative, and reliable alternative to currently used phenotypic assay for drug-resistant HIV protein. And in theory, similar assay could be developed for drug-resistant HIV reverse transcriptase, or combination assay for both of HIV protein and reverse transcriptase. And this is all for my presentation. And finally, I would like to thank the grant from Japan government and the Nagasaki University, and also thank the chair professor and the organization committee for organizing this very nice conference. And thank you very much for your kind attention.